All right, so chapter 36, resource acquisition and transport in the vascular plants. Because remember, we still have non-vascular plants, right? That's your bryophytes, your simpler plants, and then we've got our vascular plants, so the more advanced plants. All right, so I really like this picture because it's really giving you that visual of the water coming up the plants, your minerals getting absorbed, going up the plants. Then you've got the water loss through the leaves, right? Transpiration, water loss through the leaves, through the stomatas on the bottom of the leaves. Why do we have it only on the bottom of the leaves? Hopefully you're telling me it's because by only having it on the bottom of the leaves, you're minimizing water loss, okay? And then you've got the sugar that's going from the leaves down to the roots. Now, is that true all the time? Hopefully you're telling me it's not, right? That's only true when the sun's beating down and everything's great. The sugar is going from the source to the sink. But if there's no sun, then your source of sugar is going to be your root system, and your sink is going to be your leaves, because the rest of the plant is going to need that sugar, that glucose, for it to be able to transfer the glucose into ATP, right? So that's the only part, but it's trying to just give you a snapshot overall picture, so which is good. And then of course you've got the oxygen, carbon dioxide going in and out through the stomatas, which are being opened and closed by the guard cells. So I really like this picture. So please, please, please take the time to take a look at it, okay? All right. This picture right here is kind of cool because it's showing you that sometimes plants are a lot smaller than what we think, at least above ground. These right here are called stone plants, and they're found in the Kalahari Desert in southern Africa. This is the only part of the plant that's above the ground. The entire rest of it is below the ground because they only receive 20 centimeters of rain per year. That is that much rain. That much rain per year. That's not a lot. A little more. That was a little under there. So it's kind of crazy, right? So they've got these two leaves, so they can still undergo photosynthesis. They act like these little mirrors, so they can go ahead and grab sunlight. But the whole rest of the plant is beneath the ground. It's really crazy. I just think it's so cool. So we talked about all this. If you want to do a little competition amongst yourselves, a little FaceTime each other, see what you know, what you don't know. Kind of be a little fun thing to do. All right. What kind of biome would you expect to see large leaves? Rainforest, right? Why? Well, there's a lot of canopy above them. There's a lot of other plants, lots of rainfall, but what is there not a lot of? Sunlight, right? They're all competing each other for sunlight. So they're going to have the biggest possible leaves they can get to try and get as much sunlight as they can get. Water loss is not a problem for these guys. So they're like, we don't care. We have a lot of leaves a lot of leaves so we can lose a lot of water, but that's okay because it's the rainforest, there's a lot of humidity, there's a lot of water, constant rain, not constant, but a lot. So it's fine. We can lose water, but we got to get as much sunlight as possible because there's so many other plants, there's so much other vegetation. So they need to get as much sunlight as they can possibly get, so they've got these biggest possible leaves they can get. Phyllotaxy. This is really kind of cool. You're going to start looking at plants completely differently. I know I've been saying that a lot those last 2 units we've been dealing with plants, but it's really cool. All right, I'm telling you, go outside and start looking at your plants. Believe it or not, plants will have a certain degree that the first leaf and the second leaf will grow from, because they want to make sure that the second leaf doesn't overshadow the first leaf. And the actual angle is 137.5 degrees. It's going to make sure that they're going to not overlap each other. And eventually, though, you keep going and you're going to shade the lower leaves. At that point, those lower leaves will start to die and fall off. And that's why you can have this huge branch with leaves at the end of it, but nothing beneath it, because you don't want those organs, those leaves, to sit there and suck up food and not provide any kind of energy. No, sorry, not energy, but food for the plant, because it's not getting any sun. So eventually it actually performs a self-pruning situation. It's really kind of cool. Most angiosperms have alternate phyllotaxy to minimize shading. That's what this is showing you in this picture. Really, really neat. Start looking at your plants. Please go outside and look at your plants. If reduced self-shading does not occur, the shaded leaves could cause plants water. If this does occur, the plants will self-prune, like we said. The leaf orientation is vertical. It is actually better. Branching of trees, evolutionary results based on ecological niche of each species. So phylogeny, you know, not getting away from any kind of phylogeny here. They're trying to figure out how the different ways that trees have evolved. So there's multiple different thoughts right now. So we're looking at 3 different possible phylogenetic trees. That's what you're looking at here. You're not actually looking at trees. It's the phylogenetic trees of the evolution of trees. And they're still trying to figure out what it might be. So it's kind of cool. So here we're looking at grass. Obviously the leaf orientation of grass is actually better. And it's a optical illusion that that vertical blade will give you. And that's why, literally, if you look at your neighbor's grass, the grass is greener. Because when you look at it, that vertical look makes it seem greener than when you're standing above it. So when you have that angle from it, from those blades, it makes it look green. That's why grass is always greener on the other side. So kind of cool. All right. Root branching is allowed for effective water and mineral absorption and strong anchoring. Now, if you're talking about tropical storm, hurricane, do you want the tree to stand still and not give at all? Do you want a strong oak tree that's standing there with its roots strong and firmly in place? Or do you want a palm tree that's going to whip around? Start thinking about it. Start looking at what kind of trees we always see that have fallen over after a hurricane. Usually it's oak trees. They've got a strong root system, but they will stand firmly in place. And eventually, that firm resistance to the winds is what causes them to fall down. The palm trees, because their whole stem allows them, the whole trunk is willing to whip around, because they're not resisting the winds, they're giving in to a certain extent to those winds. That's what allows them to sustain those winds and not fall over. Think of your bridges, right? Our bridges, those that were resistant and strong, collapsed. Those that have a little bit of give, that's why our bridges are sturdier nowadays, do better. Same with our skyscrapers, all that. There's actually a little bit of a give. That's why if you actually stand, you can see a sway. A little sway, good. Lots of sway, bad. Just like anything. Tallest species anchored by taproots, except palms, as we just mentioned. When the pockets of low nitrogen are reached, the roots will push straight forward because nitrogen really is the one nutrient that will help plants grow better the most. Then of course you've got your mycorrhizae. Okay, this right here is, I love this little picture. You've got these grass right here growing with mycorrhizae, nice strong root system right here. That's that white that you see growing through the soil. And then you've got nice green grass blades. Then here, you can see there's not as much mycorrhizae, the root system's not as strong, but try to push through, try to get to that pocket, and the blades are much smaller, not as green, not as thick, not as healthy. 80% of land plants have a symbiotic relationship with these guys. Mycorrhizae, fungal relationship. Go back and look at that in your fungus chapter, if you please, okay? All right, now, transporting of things through the vascular system, okay? You've got apoplast and you've got the symplast. The apoplast is where things are going to go through the cell walls, extracellular space, and the interior of dead cells. Okay, it's going to go through the membranes, right? It's going to go all the way through. And then you've got the symplast. These guys are just going to go through the cytoplasm of the living cells. And it's going to be a specific channel that connects all of them through. Okay? It's kind of weird. The apoplastic, I'm so sorry, I just showed you guys the top one and that was your transmembrane route. I apologize. The apoplast is just sticking right up here. It's going through the cell walls. Okay? The extracellular space, that's your apoplast. And then you've got the symplastic route going through the cytoplasm between the different cells. Okay, so apoplastic, that's the dead part, the cell wall, and then the symplastic, it's simpler, it's going through the cytoplasm. okay? And it's just two different ways of transporting of nutrients and sugars throughout the cells. All right, now of course, your apoplastic water solutes are going to be moving around. Symplastic water solutes is going to go to the cytosol, and then your transmembrane, they're going to go one cell, then to the cell wall, then they're going to the cell again, then to the cell wall again. That's transmembrane, through the membranes, okay? Now you know if we're talking about transport, we're going to have to talk about active and passive transport. Let's do a quick review of active and passive transport. I'm hoping you remember this from Bio 1, okay? So, I don't know why I just did a graphing now. Hill, we're looking at the hill. We got a rock at the top of the hill. We got a rock at the bottom of the hill. If I let go of this rock, it's going to roll down on its own. Am I giving any energy into it? The answer is a big fat no. So is that active or passive? Hopefully you're doing this on your own piece of paper with me. This is passive transport. All right? No energy is given. You're going from an area of high concentration, remember the brackets mean concentration, to low concentration. Okay, there are three types of passive transport. You have diffusion, osmosis. Oh, osmosis is supposed to be what color? Blue, because osmosis is diffusion of water. And then you have facilitated diffusion. I'm putting a big fat star by that one. We'll say why in a minute. All right, opposite end. We've got a rock at the bottom of the hill. We want to get to the top. Am I going to have to put energy into it? Yep. So, energy is needed. So is it active or passive? It is very much active transport. This time we're going from low concentration, remember brackets mean concentration, to high concentration. We are going against the concentration gradient. And here we're going down the concentration gradient. All right, 4 types of active transport. Protein pump, protein channel, I know my handwriting's getting bad. Protein pump, protein channel, endocytosis, and then exocytosis. Okay, facilitate diffusion, protein pump, and protein channel. I'm putting a star by all of them. What do they all have in common? They all use proteins. Okay? All right. Same types of pumps and transport proteins are being used with plants as they are with animals. Nothing new. Hydrogen plays a primary role instead of sodium. Okay? Short distance transport of water across the plasma membrane. Very, very important. You guys remember co-transport? Remember I was telling you how cool it was? You pump out enough protons, aka hydrogen ions, right? Protons, just proton. You pump enough protons out, and then it was so ginormous, the concentration was so strong that it could bring in that ginormous sucrose molecule in with it through the membrane. That's what protons, that's what the protons are doing here in plants, all right? They'll do that with nitrate, they'll do that with sucrose still, they'll do it with, no, that's different. So, it's just really cool. Okay, co-transport, look at that, it's still here. I love co-transport, it's so cool. Water potential, physical properties that predicts the direction in which water will flow. You remember our 3 little beakers with hyper, hypo, iso? It's coming back. Oh, look at that. It's like stuff in Bio 1 comes back. Okay, so the reason behind the why of hypo, hyper, and iso is water potential. Okay, it's called psi. That little funky figure right there is called psi. It's measured in a unit of pressure known as megapascals. All right. I'm not asking you to know that formula. I am not asking you to. It's just know that it deals with water potential. All right. I'm going to erase this because we got to do our hyper, hypo, iso. Again, piece of paper out. Start drawing them. Start seeing how much of that you remember, because you're going to want to be able to remember it. 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. And 1 too many right there. So, okay. There. That looks better. Okay. Which one of these is a hypertonic solution? Think of it. If you gave one of these, if those dots represent sugar, which one would make the kids hyper? Hopefully you said this one. Hypertonic solution, right? If this is hyper, what's this one? You're probably going to say iso, but it's not iso, right? Just because it has 3 sugar molecules and this one has 3 sugar molecules, that's still not the same concentration, right? If I have a gallon of tea and a cup of tea and I add a teaspoon of sugar to them, is my gallon of tea going to taste as sweet as my cup of tea? No, right? That's right. The concentration is not the same. So that's your hypo, okay? That's your hypotonic solution. And then that leaves you with this one with iso. The concentration is the same. Okay? All right. I'm not using red, I'm not using red, I'm not using purple. I'm using brown. There. All right. So, if I have my cell in a hypertonic solution, we're going to skip hyper. We're going to go hypo. In a hypotonic solution, the cell will swell like a big fat hippo, right? So the cell is going to swell because water is going to go rushing into the cell. You agree? So water in. If water rushes into a hypotonic solution, what's going to happen in a hypertonic solution? The cell will shrink. Cell will shrink, water out. So then isotonic. What's going to happen to the cell? The cell stays the same. So does that mean there's no water movement? Nope. We have to have water movement. Otherwise, that's cell death. But water in equals water out. Good. All right. Let's click. Okay. So keeping these 3 things in mind. Water, pure water equilibrium, everything's great. All of a sudden we add a bunch of solutes here. So this is a hypertonic solution, right? We're going to have a problem. The water is going to push and it's going to try and equal it out. So it's going to go from here. So remember that's the U2, okay, not U2, Y-O-U-2, but U2, the letter U. And the water level is going to defy gravity and be below this water level because there is that push for the water potential to be the exact same, for there to be equal concentration, and that water potential is going to push the water to be higher. Now, if you sit here and we put positive pressure, you can have the water potential go higher in this way. Now, what happens if you have solutes and you do positive pressure? Then you can keep them the same. It's not liking it, but it can handle it. Okay? And then you can do what we call negative pressure, where you're sucking it up. And even though these two are both pure water, there's no solutes in it, so there should be no reason for the water to go from this area to this area. Because that negative pressure, you're sucking up the water. Oh, kind of like a straw, right? This right here is explaining how trees are able to suck up water all the way up from the root systems to the very tall, 200-foot tall trees, just like we do in a straw, right? As long as the straw is the right size. Okay. our cells have to be nice and plump, right? If our cells, our plant cells, are not plump, if they're not full, right? What part of the cell has to be full? What organelle? Are we saying vacuole? Good. Cell vacuole, right? The vacuole's got to be full of water so that it can put enough pressure on the cell wall to keep the plant standing nice and tall, right? That's what that looks like right here. That's a turgid plant. That's nice, plump, full of water. That's why you gotta water your plants every day. When you don't water your plants every day, your vacuole is not gonna be full of water, and it's gonna pull away from the cell wall. The cell membrane pulls away from the cell wall, right? Plasmolysis, remember that word? If you don't, I need you to. Plasmolysis. That's what's going on right there. The cell membrane pulls away from the cell wall, and it starts to shrivel up a little bit. And then your plant starts to wilt. That's not what we want for the plant. Because if the plant's wilting, are those leaves standing up nice and tall to get to the sun to do photosynthesis? The answer is no. That's not okay. This is okay. That's not okay. All right? Cells would be turgid. Loss of turgor is wilting. Aquaporins, oh, we love those, right? They're the ones that allow water in and out, or proteins that allow water in and out. Transport proteins allow water to pass through the plasma membrane. Remember, plasma membrane, cell membrane, same thing. They affect the rate at which water moves osmotically, osmosis, diffusion of water, remember, across the cell membrane. All right, 9 more slides, guys, hang in there. Diffusion in giant redwood would take centuries, okay? So we have to have a fast way to move water from the roots all the way to the top. Bulk flow, that's where bulk flow comes in. Movement of liquid in response to pressure gradient, that's that negative pressure, from high to low, and tracheids and vessel elements in the xylem and the phloem. Since there's no cytoplasm, the drain isn't clogged, and the water moves way faster. And it whoop, shoots all the way up. And I really hate this because I had all these cool pictures and actually, in theory, a root to show you guys. But I'll see if I can put the pictures on Canvas and Google Classroom. We'll see. It definitely Canvas for you guys to look at, okay? Including a really, really cool cross-section of their tree ring. It's really, really cool. So, I'll put that on there. Epidermal cells at the root tips, the hairs, are permeable to water. They'll have active transport to get the concentration minerals into the roots. You've got to suck them in. Water minerals have to pass the endodermis, that's the innermost layer of the cells in the root cortex, to enter the xylem. So here we go. You've got the apoplasmic route. Remember, that's through the cell walls. And then the symplasmic route, that's through the cytoplasm until it gets itself to the vessels. And remember, the vessels are make up the xylem, which carries the water. Oh, look at that, VWX. We always say we love the alphabet in biology, right? Endodermal cells have a Casparian strip, a waxy material that forces water nutrients to enter the cells via the plasma membrane instead of through apoplastic route. So this way, it's able to go through faster. Root pressure. Root cells pump ions into the xylem of the roots. Water flows in through the osmosis, creating a positive pressure. It's going to push materials up the xylem, so it pushes it up. Transpiration, cohesion, and tension mechanisms when all of them work together. So the loss of water through transpiration through the stomatas, that's what allows that negative pressure to occur, sucking up water up through the xylem, so that water will keep going up. When the stomatas are open, it evaporates from the spaces out of the mesophyll and infuses out the stomata. Water is replaced in the airspace by evaporation from the cell walls. This creates a negative pressure and sucks it up. So it's just about, you've got the positive pressure coming in, and then you've got the negative pressure pulling them up. So you've got the positive pressure putting everything into the cells, into the roots, and then you've got the negative pressure pulling them up through the stems to the leaves. The combination of the two working together is how plants drink. The pull and push of water is felt throughout because water is cohesive, right? Couple, co-workers, it's water to water bonding together. Look at that. That's literally going back to our very first foldable, you guys. This combination of push from the roots and pull from the leaves and stickiness of the water itself creates this constantly moving column of water up through the plant. So cohesion, negative pressure, loss of water, and positive pressure from the water getting shoved into the roots, all of it together, is what allows the plant to drain. Transpiration is evaporation of water through the leaves of plants, these stomatas. Leaves have large surface area, but they're super thin, which provides large surface area to volume ratio. This allows for a high rate of photosynthesis and water loss. To control water loss, stomatas are used, right? We open and close them depending on what we need to do. Stomatas openings which are surrounded by specialized cells called the guard cells. This is nothing new. We know all this. That's why you're going to have big, ginormous leaves in the rainforest where there's lots of water. We can lose all the water in the world. That's not a problem. But then in the desert, you're going to have those spines that have much thinner, those specialized leaves in order to minimize water loss, because obviously that's your number one problem. And then you're only going to open stomatas at nighttime. Guard cells, bean shaped. You're going to see really cool pictures of those guys on the lab. Really, really cool stuff, okay? Guard cells are right here. Bean shaped with thicker cell walls next to the opening in most angiosperms. Thicker right here. That's a thicker cell wall. Why? When the guard cells is full of water, turbo pressure pushes on the weaker cell walls away from the opening. So it'll go ahead and open it up. Potassium is also involved in this. Guard cells will actively transport potassium in and out in order to go ahead and create the proper gradient they need to either open or close the stomatas. Typically, the stomatas are open during the day and closed at night to prevent water loss when photosynthesis is not occurring, because why would you lose water if you're not going to undergo photosynthesis? 3 more slides. Translocation, the product of photosynthesis being moved via the phloem. The phloem is made of sieve tubes. Remember, sieve tubes made up of phloem, which carries the sugar. One of the few times I'm okay with you using sugar instead of glucose. Direction of move in a phloem sap can be up or down. Remember, we said that. Direction can change, but it always goes from sugar source to sugar sink. Source to sink. Alright? Sugar source, organ that is net producer, usually the leaves. The sugar sink, organ that consumes or stores the sugar. The growing areas, roots, buds, and fruit. Don't forget your fruit. That's why fruit tastes so sweet. In the summer, the cells that store the sugar can be sugar sinks when the photosynthesis is so high. Phloem sap moves by bulk flow or pressure flow pushed by the high pressure of the source cells. All right, guys, that's it for chapter 36. Good job.